Hello, my name is Joshua Mugabe. I welcome you to DNA. DNA stands for deoxyribonucleic acid. DNA stands for deoxyribonucleic acid. And on the side here, we have a section of DNA. This is a section of a chromosome with very many nucleotides, bases, sugars, and phosphate, forming a DNA molecule. This chemical substance is present in the nucleus of all cells of all living organisms. Deoxyribonucleic acid is present in the nucleus of all cells of all living organisms. DNA controls all the chemical changes which take place in the cell. All the chemical changes that take place in the cell are controlled by DNA. The kind of cell which is formed, for example, the muscle, blood, nerves, is controlled by DNA. So whichever kind of cell that is going to be formed is going to be also controlled by DNA. All the chemical changes that are taking place within the cell, they are controlled by DNA. The kind of organism which is produced, for example, buttercup, it's a giraffe, a herring, human, etc., they are all controlled by DNA. So, still we are seeing that any type of organism that is formed, whether it is a buttercup flower, it is a giraffe, which is an animal, it is a herring, which is fish, it is a human, every organism that is produced, the production of that organism is controlled by DNA. DNA is a very large molecule which is made up of long chains of subunits. So when you look at this part here of a nucleotides, it can form the DNA, but it is made up of very many subunits. When you look at it, it's made up of very many subunits. The subunits are called nucleotides. The subunits are called nucleotides. Let us look at the nucleotide here. Now, for example, if you look at this section of a chromosome, we have what we call the sugars, the pentose sugar. This one is a 5-sided sugar. It is one subunit. It has got what we call the bases. Here we have cytosine is a base, adenine is a base, guanine is a base, thiamine, all these ones are subunits. These are bases. Then we have the phosphates. The phosphates, these ones are also subunits. So when we have a phosphate, we have a sugar, and we have a base. That one is referred to as a nucleotide, and it is a subunit. So the DNA is made up of long chains of subunits called nucleotides. Each nucleotide is made up of a sugar, and this sugar we can call it deoxyribose sugar. So this one is our sugar, the one we are calling deoxyribose sugar. So every nucleotide is made up of a sugar, small h unit. That one is the sugar unit, and it is called deoxyribose. Then another subunit is the phosphate group. The phosphate group is now this one here. This is a phosphate group. It is another subunit of a nucleotide. That is a phosphate group. A phosphate group. Those are phosphate group. And also the organic base. It's made up also of the organic base. Now the bases are these ones that we have in the middle. These are bases. Guanine here, and then we have cytosine. Those are bases. Adenine and thymine. Those are bases. Cytosine and the guanine. All those are bases. So a nucleotide is made up of a sugar. For example, this is a sugar, which is called deoxyribose. It's made up of a phosphate. This one is a phosphate subunit, and it is made up of an organic base. So all that one is now what we refer to as a nucleotide. This one here, a base, a sugar, a phosphate, and then here that one is called a nucleotide. So DNA is made up of very many chains of nucleotides. Ribose is a sugar like glucose, but with only 5 carbon atoms in the molecule. So ribose is a molecule, but it has only 5 carbon atoms compared to the glucose. The glucose has got 6 carbon atoms. Glucose is a 6-sided figure, and ribose is a 5-sided sugar because it has only 5 carbon atoms in its molecule. Deoxyribose is almost the same, but it lacks 1 oxygen atom. So deoxyribose resembles ribose, but the difference between ribose and deoxyribose is that deoxyribose lacks 1 oxygen atom. The most common organic bases are, we have the most common organic bases. The first one is adenine. This one is adenine, is one of the bases and is represented by letter A. Then we have another organic base, which is thiamine. Thiamine is this one, and it is represented by letter T. The third one is cytosine. Cytosine is this base, which is represented by letter C. Then the last base is guanine and is represented by letter G. Those are the most common organic bases that are subunits of the nucleotide. Deoxyribose, the phosphate, and one of the bases combine to form a nucleotide. So we have the sugar, which is deoxyribose sugar. and then we have the phosphate unit combining together with one base like adenine. All of that one, they combine together and they form what we call a nucleotide. We have a phosphate, which is this one. We have a sugar, which is deoxyribose, and then we have adenine, which is a base. When they are combined together, they form what we call a nucleotide. The phosphate group gives the DNA its acidic property. This phosphate group is the one that gives the DNA its acidic property. A molecule of DNA is formed by millions of nucleotides joined together in a long chain. So when we talk of DNA, DNA is made up of millions of nucleotides, and these nucleotides are joined together, and when they are joined together, they will form a longer chain, and now that long chain can be referred to as a DNA molecule. A DNA molecule is made up of millions of nucleotides joined together, and when those millions of nucleotides are joined together, then we form what we call a DNA. Remember, we have said that a nucleotide is made up of phosphate joined to the sugar, and the sugar is joined to a base to form what we call a nucleotide. And a nucleotide will join with another nucleotide, and join another nucleotides, and they form long chains of joined nucleotides, which will form now a molecule of DNA. In fact, DNA consists of a double stranded of nucleotides. It consists of a double strand of nucleotide. So here we can see there is this strand of nucleotides, and then there is another strand of nucleotide this time. So DNA is made up of double strand. It is a double stranded. So we have one strand this side. This is one strand of nucleotides. Then we have another strand this side. This one is another strand, and both of them, when they are joined together, we call it a double strand of nucleotide, which forms the DNA. The sugar phosphate chain are on the outside of the strands and are held together by chemical bonds between the bases. So what we call as the outside strand is this one here, which is made up of the sugars and the phosphate. So the sugars and the phosphate, these are the sugars and the phosphates, they are on the outside. They are forming a chain which is on the outside of the strand. The same applies on this side. This side we have the phosphates and the sugars forming a strand outside part of the strand. Now these two strands, this strand and this strand, they are held together by a chemical bond between the bases. Now we have a bond there, that is a base and a base bonding there. We have a bond here, we have another bond Those are bonds, and those bonds are between the bases. The bases form the bond. For example, here we have a base and a base, they form a bond. Base and base, bond. Base and a base, they bond. A base and a base, and bond. So those bonds which are between the bases are the ones that are holding the strands together, which we call the double strands of nucleotides. The bases always pair up in the same way. The bases are always pairing up in the same way. For example, if you look here, we have these bases pairing up. Adenine is pairing up with thymine. And again, we have other bases here. We have cytosine is pairing up with guanine. So they will pair up the same way like that. Now, when you come here, you have, this is thymine pairing with adenine. Then here we have a guanine pairing up with cytosine. So that's how they pair up the bases. Then we say that adenine, the adenine base, form a bond with thymine. As we have already seen up here, adenine forms a bond with thymine. Then we have cytosine bonds with guanine. Like the way we have seen it here, cytosine is going to bond with guanine. The paired strands are coiled into a spiral called a double helix. Now we have two strands, and these strands, we are calling them the paired strand. There is this strand here which is coming like this, and then there is a second strand here which is coming like this. These strands are joined together by the bases. We have bases which are joining the strands together. Now these bases, they join the two strands together, but this strand, now the double strand, goes on forming a coil. It coils itself around, and that spiral is what we call a double helix. Double helix. Here we have a DIY model. This is a DIY model, part of the DNA molecule. And this is a teaching model. It is just a teaching model, but the colors that are on the bases do not conform to the accepted scheme today. So here we have our helix strand of DNA. As you see, it goes on coiling around. We have on the sides here, we have the phosphates with the sugar molecule forming the strand outside, and then we have the bases connecting in the middle. The bases connecting in the middle. Cytosine connecting with guanine, thymine connecting with the adenine. So that's how they connect. Before a cell divides, the DNA strand unwind and separate. So previously we have talked about cell division, but before cell division takes place, the DNA strand will unwind and separate. For example, here you are seeing the two strands have been separated. The strands have separated. That is during the unwinding, and then they separate. That is when the cell is undergoing cell division. Now each strand will make a new pattern by adding appropriate nucleotide. Now, this strand here is going to make other new patterns by connecting and forming other, connecting to other nucleotide. We shall have a base here, and then the base we shall have another sugar here connected here, and then we shall have a phosphate. So it is going to go on picking bases connected with sugars and then a phosphate, and then it will form another strand here. It will form another strand here, which is going to connect to form a new pattern. The same applies to this one. This one will also connect to a base and then another sugar and then a phosphate. It will go on connecting like that, another base, sugar, and then a phosphate. So these ones will connect together again and they form another pattern or another strand of DNA, connecting themselves like that. So when they connect it together, that is what we are saying, that each strand will connect and form a new pattern by adding appropriate nucleotides. Now we are seeing here that as a result, there are now two double-stranded DNA molecules in the nucleus. This one has formed its pattern. This is its pattern now. And the other one has also formed its pattern, which is this pattern here now. Now we have two double-stranded DNA molecules in the nucleus. In the nucleus, we had one-stranded DNA molecule which unwinded and then separated, and then they started forming patterns by joining different nucleotides. So now this one will become a double-stranded DNA molecule, and then we have another one also here, another one here, which is also a double-stranded DNA molecule. So in the nucleus now we have two double-stranded DNA molecules in the nucleus. So that when the cell divides, each nucleus will contain an identical DNA. Now, we was taking this one to be a cell, and this cell was having, maybe let us say, having one double-stranded DNA strand. But now after dividing, and then the cell is going to divide, therefore the cell, what is going to happen to the DNA, it will unwind and separate, and then it forms a pattern. Then we shall have two now DNA in the cell. So when this cell now divides in the middle, each cell now will be having a double-stranded DNA molecule which is identical after cell division. This process is called replication. The process of forming patterns, which was one chromosome and they become two chromosomes, that one is a result of replication. It is called replication, whereby the chromosomes form, they double themselves and form patterns. The sequence of bases in DNA forms what we call the genetic code. The sequence of bases in DNA, they form what we call a genetic code. Now, a group of 3 bases, which we call triplet, they control a production of a particular amino acid in the cytoplasm. So we are saying that the sequence of bases in the DNA, they form what we call a genetic code. What we are referring to as if you have 3 bases next to each other, that one is what we call a genetic code. For example, we have cytosine, adenine base, and thymine base. They form a code, a genetic code for valine. And if we have cytosine, guanine, and adenine, these ones, they form a code which we call a code for alanine. So the group of bases, 3 bases joining together, these 3 bases are the ones that we are calling a triplet. And they are going to control the production of an amino acid. So this triplet here, which is cytosine, adenine, thymine, it is a triplet which we call a code for an amino acid called valine. And if we have another triplet, which is cytosine, guanine, and adenine in this order, CGA, they are going to form a triplet which code for alanine amino acid. The different amino acids and the order in which they are joined up determine the sort of protein being produced. So if we have very many amino acids and the order in which they are joined up together will determine the protein which is going to be produced. This is known as a triplet code. A triplet code, we say, if we have 3 bases, those 3 bases, they form a code. And that code is called a triplet code. Here we have cytosine with cytosine and adenine. That is a triplet code. And here we have guanine with adenine and guanine. That is a triplet code. That's what we call a triplet code. And each triplet code for a specific amino acid. So every triplet is coding for a specific amino acid. For example, we have here cytosine, and adenine. This is a triplet and it codes for glycine amine. It codes for glycine amine. We have here guanine and guanine bases, which we call a triplet. These ones are codes for an amino acid called proline. So every triplet codes for a specific amino acid. The amino acids are joined together in a collective sequence to make part of the protein. So we can see that these amino acids that have been formed are now joining together to form a particular amino acid. But this amino acid has been now formed. These amino acids have been arranged together by a code which is in the DNA. So that code is the one which is going now to arrange the amino acids together. And when the amino acids are arranged together, they will join and then they will form part of the protein. So you can see that this protein that is going to be formed here is controlled by these different triplets joined together. And these triplets, we are finding them in DNA, which is found on the chromosome. This is a small imaginary protein molecule showing how the sequence of 5 different amino acids could determine the shape and the identity of a molecule. So here we have 5 different amino acids: serine, cytosine, valine, glycine, and alanine. They are joining themselves together and they are forming a particular shape. The way they join themselves together, they form a particular shape and the identity of a particular molecule of a protein by just joining 5 different amino acids together. The protein builds up cell structures. So when we have got the amino acids and we have joined these amino acids together, when we join them together, we end up forming a protein. And this protein now, which is formed, is going to form a particular cell structure. They also make enzymes. Proteins are the ones that are going to form enzymes that are going to be used in the cells. And the DNA controls which enzymes are made. And the enzymes determine what reaction takes place in the cell. So we are seeing that the enzymes which are going to determine the reactions that are taking place in the cell are made by the control of DNA, because DNA is the one which is going to make the protein which has made the enzyme, and the protein is coming from the amino acid, and the arrangement of the amino acid is determined by the triplet found in the DNA. These triplets are coming from 3 bases which are found in the DNA. So the structures and the reactions in the cells, they determine what sort of a cell it is and its functions. So it is the structure and the reactions that take part in that particular cell that will determine what sort of cell is that and its function. So you can see when we say that the DNA determines the structure, it determines the reaction, and it determines the sort of cells formed and the function of the cells, all of them are controlled by the DNA molecule. So DNA exerts its control through the enzymes, because the enzymes are the ones that are going to control the reactions that are taking place in the cells. And it is these reactions that are taking place in the cells that are going to determine the type and the function of the cell. So the DNA is going to exert its control through the enzymes that have been formed. So a sequence of triplets in the DNA molecule may code for a complete protein. So these are the triplets. These ones are found in DNA, and they are the ones that are going to code for a particular protein. This is a part of the protein that has been coded by the triplets that are found in the DNA. Such sequence form a gene. Such a sequence will form a gene. There are many, there may be thousands or more bases in one gene. So in a gene, we may find there are very many bases. In one gene, it might be having very many genes. They might be having very many bases, and a gene is going to produce a particular protein which is going to determine a particular character of an organism. Thank you very much for participating in DNA. Please do like, share, and subscribe.